Our next speaker is Dr. Hanlee G. Dr. G is an associate professor of medicine here at Stanford. He's a practicing oncologist. His laboratory focuses on developing novel genome sequencing technology. He has a particular focus on gastric cancer. He's the PI of the Gastric Cancer Registry, which is a North American registry and biospecimen repository of gastric cancer. He's also my mentor, and today he's going to be talking about molecular pathways of gastric cancer and intestinal metaplasia, insights from single-cell transcriptomics.
I'd like to thank Robert for the introduction, also for the organizers of this meeting, and it's a real pleasure to see so many members of NCI and the rest of the community. So I need to come to these kinds of meetings more often because I'm really enjoying it. The overview of the talk has three components. Number one, I'll give some brief description about single-cell genomics, which I think is going to have potentially revolutionary implications for translational sciences and for gastric cancer detection among those topics. I'll describe a pilot study of gastric metaplasia and carcinoma, which I think will prove hopefully to be fruitful in terms of future genomic and what I'm terming cellular biomarkers of progression. And then a novel system where I think there's an opportunity to really go beyond just simple observation and consider perturbation studies where we can actually define phenotype of even individual cellular populations that exist within the microenvironment surrounding some of these lesions and primary cancers. So the topic, cancer and metaplasia. As you know, and as we've seen from multiple photographs and demonstrations of pathology and all these wonderful talks, is that gastric cancer is amazingly complex in its cellular structure. And I think when I oftentimes talk to my genomic colleagues about their analyses, is that once they've had the opportunity to actually see these things either on pathology, gross, or imaging studies, the sheer challenge of trying to understand how these tumors behave or how they even develop seems inordinately high. So this is just a demonstration of a resection, as you can imagine, and follow up using multi-parameter immunohistochemistry to look at some of the explicit cellular features. Well, I think the complexity is clearly displayed by this matched normal tumor or matched dysplasia example, where all of these cellular features, as we see right now, it's very difficult to account for the underlying biology that might distinguish particular pathways, that might even preclude or identify metaplastic lesions that will go on to progress. And so when you consider the current methods that have been used up to date, most of them involve taking these types of features and basically grinding them, averaging out the molecular signatures. But with the onset of single-cell genomics, and particularly now that systems are reaching the point where we can do millions of cells within a matter of days, our ability to delineate this complexity at individual cellular level, looking at explicit molecular transcript features, DNA features, epigenetic features, is increasingly growing. So in terms of looking at potential genomics of gastric metaplasia, I'd just like to cite the work of Dr. Nuja Satha, who's the first author on a paper and the lead researcher who worked with Robert and George Poutsides, a very close colleague in surgical oncology at Stanford. Really the emphasis of this translational study was to leverage single-cell sequencing on particular low cellularity biopsies. Now, one advantage that we had is, albeit we're still getting some of our surgical resections from Dr. Poutsides, which are relatively large, the ability to do traditional types of single-cell analysis through flow cytometry is oftentimes restricted because you're dealing with hundreds of thousands of cells minimum. But when we use some of these single-cell platforms, we can examine the number of cells down into the thousands. And so for an endoscopist like Juhan Robert, that means an accessibility to samples that you previously couldn't analyze. In particular, we have the ability to look at aspects of the gastric microenvironment and immunologic factors, as well as other types of cellular populations that previously you really couldn't identify, and discretely at individual cell level. And with that, you can increasingly look at very explicit and specific cellular genomic alterations at this resolution. So among the advantages of single-cell genomics that I already described is we can look at transcriptional alterations among these complex cellular mixtures, really identify rare cell types that would be otherwise nearly impossible to see with methods such as immunohistochemistry or flow cytometry. We can consider the stochasticity of gene expression, which has its own complex regulation, but we can see these kinds of events. And we can look at specific pathway alterations in addition to differential expression among specific genes. And that allows us to really consider what we're terming cell states. What is the state of a cell within a given microenvironment, whether it's metaplastic tumor, dysplasia, or normal gastric tissue? So the structure of the study involved taking biopsies with tumor metaplasia as well as paired normal that was typically several centimeters away from that given initial biopsy site with a suspicious lesion, dissociation to single cells, and then subsequently single-cell RNA sequencing. In addition to really explore some of the immunologic variation, we had a small subset of samples where we had peripheral monocytes available, and we also simultaneously subject those to single cell. The platform we used is the 10x Genomics platform, and what that involves is taking individual cells and partitioning them into oil emulsions that get incorporated with a cellular barcode, so each cell can be subsequently identified. As part of that process as well, we're able to identify and sequence individual RNA transcript molecules, which really gives us pretty high resolution to differentiate expression. And just as one of the latest things that my group has developed, we can do complete transcript sequencing and use that as a way to look at rare allelic variants that may occur in specific subpopulations. We're very excited about that. So half of my group is dry lab computational, and we have the benefit of people like Sue Grimes, who's one of our bioinformatics analysis, to develop automated pipelines such that we can rapidly take this single-cell sequencing data and post within a matter of a day or so, generate a whole series of readouts that include looking at differential expression from individual cells, looking at trajectory based upon RNA transcript status that gives us some sense about which cells may be going into or out of specific cell states, looking at the regulation of cells based upon their presumed transcriptional factors, looking at cellular interactions in which we can consider ligand-receptor interactions based upon the strength or the level of expression of complementary genes that encode these things. We can also identify copy number grossly from individual cells using RNA expression that's averaged across large segments of chromosomes, and use this to really begin to do cell type assignments. We're employing things like machine learning and whatnot to really say this is a cell in a given state that's attributable to a specific cell type. So for this first study, we did 7 tumors and 1 metaplasia sample, as well as the matched normal and PBMCs using high-throughput single-cell RNA sequencing. Underwent a series of rigorous quality control computational methods in order to get at the data. Again, most of this is automated. And then used a series of these to produce what's typically known are U-maps, where you see 2 axes and then every individual dot represents an individual cell. The clustering and general proximity using U-map is indicative of cell types that have very similar transcriptional features. And then all these additional readouts were provided. So one of the most striking things we saw from the analysis of epithelial cells as part of this pilot study was the fact that we saw the full repertoire of normal epithelial cell types that were representative of all the different sources, the ones that are located within specific portions of the glands and whatnot. Most interesting and relevant is there were 2 distinct cells that we termed GC type 1 and GC type 2 for a lack of a better descriptor. And you can see that based upon this UMAP description where these individual normal tumor cells are represented by individual dots shown here that were aggregated across the entire sample set. Metaplasia samples are shown by these purple dots which seem to aggregate here. And then the normal epithelial cells were distributed amongst different clusters, oftentimes indicative of different types of function. We found clear evidence of normal epithelial subtype markers that have been typically cited in the classic literature using immunohistochemistry or flow, and these are a couple of examples where we can find those. So we know that the data was relatively representative of what's been assumed to be canonical features that you'd see among normal epithelial subtypes. I think more importantly is that when we begin to look at this, quote, carcinoma or GC type 1 versus GC type 2, actually there's a little error there. GC type 2 was associated with the metaplasia and the carcinoma was associated with the type 1. When we look at these gene pathways, we can take these individual gene differentials among singletons and then aggregate that and look at the overall activity of explicit pathways. And so using that type of information, you can begin to see that there's some clear dysregulation that's starting to happen among some of these cell lines, among some of these individual cells, The majority of the metaplasia cells came from the gastrointestinal metaplasia sample. So this enables us really to start defining some of the pathways that are aberrant and perhaps begin to consider making predictions about intestinal metaplasia cells that may be more prone to going on towards becoming full-fledged invasive carcinoma. Differential expression also was very explicit in that we could see there were a number of genes which are highly differentially expressed that seem to separate, say, carcinoma from the small pilot set versus metaplasias. And so some of the logic that I had when we were setting this up with Anuja was the fact that we could use the invasive carcinomas as kind of an aggregate data set to draw conclusions about the common things that might be differentially expressed, and then see amongst our metaplasia samples ones where they showed similar differential expression or alterations in pathway activity that may be indicators that that particular lesion, that GIM, is progressing. Copy number variation turned out to be a very convincing data set in order to differentiate individual cells. So what you're looking at here are representatives of the individual patients. So there's 2 patients, one with identified histopathologically confirmed gastric cancer. And you can see that we identify the different epithelial cell types here represented by this color coding. And then subsequently, each time you see one of these dots, it's indicative of a copy number change here. And the individual copy number changes are ordered based upon their characteristics, going from chromosome 1 down through chromosome 22. The individual columns that you see represent individual cells. So you can see here that there are clearly clusters here from this where there was a deletion in 4q and an amplification in 20q, and that's oftentimes seen in chromosomal instability. Within the metaplasia sample, there were a number of cells that showed some of these indications where there was copy number changes, including an example of several cells that are showing an amplification at 5q and whatnot. So I think that this presents an opportunity where we can not only grossly consider the overall averaging of instability, genomic instability from metaplasia, perhaps use that as a quantitative metric, but also identify more explicit copy number changes or changes in chromosome state among metaplasias that could be indicators of progression. Well, we also do single-cell DNA sequencing within the group, and we know that we can leverage those types of technologies in addition to RNA-seq to provide much higher resolution definition of some of these instability events. And this represents a gastric cancer tumor in which we have a set of approximately 10, including some colorectals, where we're defining individual copy number changes using whole genome sequencing per given cell to delineate some of these small points. Now these kind of discovery modes can be relatively expensive. They can't be translated immediately into exact biomarkers, but we do have systems where we could take, say, 1 million cells or 10,000 cells and be able to target individual regions which are more predisposed to instability changes or transcriptional changes, and use that as a way to identify discrete markers, drive down the cost of doing these single-cell analyses so that we can explicitly monitor these people. The tumor microenvironment is an amazingly hot topic right now, and I think it's a recognition of the overall complexity that we see in these lesions. So this is a plot where Anuja looked at the various cell types that exist, and you can immediately see that we see all the variety of cells that we would expect to see among the variety of patients. So in this particular graph, you're seeing the fraction number of cells across a series of different cells, where you're seeing black is representative of epithelial cells, orange indicates cancer-associated fibroblasts, T cells, N cells, B cells, endothelial cells, tumor-associated macrophages. So again, we get this beautifully quantitative resolving picture of the overall microenvironment composition of these given biopsies. And again, we can do this on endoscopic samples. And so similarly, tumor-infiltrating lymphocytes, I think, represent an important cellular readout. And you can see amongst these tumors that you see that there are a variety of different cells, immune cells, that we can clearly delineate using these methods, including black noting naive CD8s, CD4s, natural killers. And so my hope is that with these types of readouts, we can get away from considering one-offs where we're using a handful of proteins for IHC and start considering cellular readouts that are more taking in consideration number and types of cells, whether they be stromal, epithelial, or perhaps even immune-based lymphocytes that could be indicative of certain type of inflammatory processes that are more likely to predispose to development of a full-fledged invasive carcinoma. So this is just a brief example where if you look at our metaplasia sample and the absolute number of cytotoxic T lymphocytes, within metaplasia we saw a reduced number. Within the normal tissues we see these kinds of numbers. And then we see interesting shifts among the tumors where this cluster 0 had a significant increase of these individual cells compared to the other samples. And from that, we can actually cull out aspects of the transcriptional regulation of those cell types as well, as demonstrated by this heat map here on the side, where the individual cells are represented by columns, and genes that show significant dysregulation per gene expression levels that have been corrected for multiple testing are shown in addition. So the patterns are quite clear when you begin to look at them. Now, single-cell analysis in my dreams eventually will be something that's routinely done on all clinical samples. I know that there are hurdles there. I am not worried about the sequencing cost. That is dropping like a rock. But there are many, many samples in which that can't be subject to. So the way that I see in order to translate these single-cell findings is to use methods such as in situ probes to validate them. So as an example of this study, in order to look at the variety of different types of immune cell populations, Anuja looked and used a particular immunohistochemistry panel with fluorescent probes to define these populations very efficiently. And you can see that she's able to apply this across the entire sample set. So thinking about the single-cell data sets as the initial discovery set, you can kind of imagine that afterwards, if you have a variety of candidates, one can develop in situ either antibody-based or RNA expression-based transcript staining to be able to go back and look at archival samples to verify some of these conclusions. And from this, she was able to show that there are certain cell types that are depleted, such as CD3-based T cells. She looked at PD-1 expression, as well as a couple of other markers, again, indicating the type of immune cell changes that she had confirmed on single cell, but using spatial imaging from her histopathology samples. So just as kind of a wrap-up, immune cells and T cells are not the only thing that we've noted that are quite different. So macrophage differentiation was one of the surprise findings, where we found that if you looked at overall all the macrophages across every single gastric cancer, there are certain types that showed distinct myeloid lineages, whether they are coming from or infiltrating tumors, versus PBMCs, which are shown in green, versus metaplasia. And it turned out they have very distinct transcriptional properties that did not fit into the classic categories of macrophage function and type that have been described before. Finally, from this kind of data, you can look at aspects of intercellular communication, where you look at a given ligand's expression and its matching receptor, and you can create these network maps. And so using these kinds of inferences combined with other type of computational methods, one can consider whether there's dysregulation in the signaling that maintains a normal gastric microenvironment compared to a intestinal metaplasia microenvironment versus a tumor. And I think that this could be a great area to identify candidates to do more granular, higher throughput, less costly validation studies looking at these markers explicitly. So as the last part, I'm going to touch on work that Jay Chen did, because this is great observational data, but still it doesn't really get at the root of biology. So one thing that Jay and Anuja have been involved in is developing systems in which we can go and perturb the environment of these very complex cellular biopsies. And so the way that that works currently, and we've actually done it with one of the samples that Robert's provided, is we can take a biopsy and we can maintain that within tissue culture up to 14 days at best. That allows us an opportunity to look at that transcriptional profile at single cell resolution, look at the sheer complexity of all of those different cell types, but then also offers a chance to perturb that. So if you take a given vehicle and compare it to a control, that allows you to use a single-cell RNA sequencing comparison after a day or several days, look at the perturbations and consequences of that given additive in comparison to control. And so Jay actually did that, where he used IL-2 as a generalized way to stimulate T cells, and noted that there was a whole series of changes when he compared the metaplasia versus the normal tissue cells between the two cell types. And so that enabled us to have a functional readout where we could see explicit T cell changes being induced by the addition of IL-2 within the context of the gastrointestinal metaplasia environment versus the normal tissue meta-environment. And so he saw a couple of changes in some of these transcriptional factors and genes such as GZMB, where there seemed to be an increased transcription within the specific cell types versus the normal cells. So I think this gives us an opportunity to probe some of those cellular interactions and define some of these phenotypes based upon actual biological perturbation. So in conclusion, single-cell genomics is increasingly proving to reveal these critical genomic and cellular features of cancer and metaplasia, and thus I think it represents a wonderful opportunity to develop the next generation of cellular biomarkers, as well as identifying explicit targeted molecular markers that can be disseminated for more general validation studies. And finally, using these kinds of systems, we may have an opportunity increasingly based upon these primary samples from patients that have been affected to actually understand biology by doing these kinds of ex vivo perturbations where we can look at the molecular consequences if we affect a cellular population and see if they've been dysregulated compared to matched normal tissue. So in the future, we have technologies where we can look at associated pathogen transcripts. So for the ability for us to look at complete EBV transcripts using long-read sequencing from single cells is something that's probably we could develop relatively quickly. We're doing an expanded study for additional cellular biomarkers beyond this pilot. We're integrating in microbiome features using a variety of other sequencing modalities, and that's a study that some of the group members, including Robert, are really driving forward. And then we have advanced machine learning-based features where we can take a look at histopathology, even fundamental H&Es, to try to identify some of the dysregulation or cell types that may have morphologic changes that we can see. So I'd just like to recognize Anuja Sata and Jay Chen for their critical contributions. Sue Grimes, Billy Lau, and Allison also were critical in making the study happen. Robert has been a great collaborator and has been providing the gastrointestinal metaplasias, and then our dear colleague George Pulsides, who's also been critical in making these studies go forward in terms of gastric cancer. And here are my funding sources. Thank you.